floral diagram and floral formula. I'll tell you what you have to do. The question will be directly from the NCERT, from this chapter. So, and basically, now let me write it down what exactly they will ask you. Morphological. Fine. Fabaceae. So let's start with the Fabaceae. So as I have already told you that we were discussing, this is the formula of Fabaceae. Fabaceae used to be known as Papilionidae, right? Or we can say this is the older name of the family Fabaceae. Nowadays we are calling it Fabaceae, and there is a subfamily, means family within this family. We call that subfamily. That is a Leguminosae, and I have already told you the Leguminosae name came from the legume. Means these plants are known to have a fruit, and we call that fruit legume. You must have seen the legumes in many of the plants, like beans and all that, right? And this is the family of the pulses. All the pulses come under the subfamily Leguminosae, and Leguminosae is a subfamily of the Fabaceae. Now, what is the floral formula or floral diagram? This is the floral diagram. This is the floral formula. So this is the symbol of zygomorphic. Zygomorphic means there will be bilateral symmetry. Means you can cut this flower in two equal parts from only one axis, right? And that will be this axis, right? That's why this is called what? Bilateral symmetry, right? And this placentation seems to be marginal. This is called bisexual. This is symbol of bisexual. Male and female sex organs are both are available over here. We can see this is androcium, this one, this is androcium, and this is gynocium. Both are present. So male and female, both reproductive organs are present. That's why we can call it bisexual. Calyx 5 gamo. Look, these are the calyx. All calyx are united, right? So I have written over here, right? Calyx 5 and 5 into bracket, right? So calyx 5 gamo. All calyx are united. Now come to the corolla. Corolla 1 plus 2 plus 2 gamo. Look, this one is free, so no bracket. These two are free, no bracket. And 2 gamo, these two are fused. That's why these are gamo. There is a gamo, right? Bracket is present. So we will write like that. Corolla 1 plus 2 plus 2 gamo, right? Now, what kind of the estivation is this? I explained you in last class. What will be the estivation for this? Come on, I have told you. Standard wings and keel. Come on, guys. Don't tell me.
Vexillary.
No. Yes, very good. Vexillary estivation, very good. Vexillary estivation. We have already discussed that. Look, let me show you. Corolla, look. This is the vexillary estivation. I have told you. Upper one is standard, then there are two wings. So there is a one standard, two wings, and two keel, and these two keel will be fused. So I have told you that when the arrangement comes 1 plus 2 plus 2 gamo, it means the estivation is vexillary. Now you can see the same estivation in the Papilionidae in family Fabaceae. Look, in Fabaceae, this is the 1 plus 2 plus 2 gamo. So we can directly come to the conclusion that this is the what kind of estivation? This is the vexillary estivation. Androcium 9 plus 1. Look, so androcium, these 9 androcium in a group, and single androcium is the group. What kind of androcium this is? Tell me, guys. Monoadelphous, diadelphous, polyadelphous. Come on. Androcium 9.
Again.
What will be the arrangement of the stamen? Monoadelphous, diadelphous, polyadelphous. Very simple. Look, again, let me remind you. Androsium. I have told you if
Mono.
androsium, this diagram, look, I have told you that when polyandrous and androsium are separate, gamoandrous when androsium are united. When androsium are united in a group, we will call it, we will call it monoadelphous, right? When there are two group, diadelphous. More than two group, polyadelphous. So what is the arrangement of androsium here? They are in two group. Yes, a group of 9 and a 1, right? So there are two groups. So androsium are diadelphous. Clear or not?
Yes, sir.
Gynosium 1 gamo, ovary superior. Clear?
Yes,
Now,
sir.
what you have to cram here, what you have to remember here first. So, bisexual zygomorphic, that you know, what you have to remember, that economic importance. Economic importance. Example, you will get either from the directly question from this, either you will get this formula and you have to tell that which family belongs to this formula, right? That kind of the question. Or you will get the question from the which of the you will get an example of any plant, the name of any plant, and you have to tell the family which it belongs to, right? So there are certain examples. So pulses, I have told you, pulses, all pulses come under this family, like gram, moong, arahar, right? Glycine max, soybean, and then in dye, indigo, you know, indigo. This question I've been asked. Indigo belongs to which family? Indigo belongs to this Papiliondae family, Fabaceae family, right? And fodder. Fodder is normally given to the cattle animals, right? Cattle. So trifolium. Trifolium have already been asked. Trifolium. You will get the question. Trifolium. Trifolium is a fodder. It belongs to this family, right? Then sweet pea, right? Ornamental plant. Ornamental plant. In ornamental plant, sweet pea, that is called garden pea. It belongs to this family, and that's it. That you have to remember these examples. Clear?
Yes, sir.
Okay. What is the fruit? Fruit is legume. One more thing, remember, fruit. If someone asks you what is the fruit, fruit is legume. Is that clear? If you have any doubt, ask me. Any one of you. Right? Whatever written on the screen, that you have to, everything, every word you have to remember. Else you will miss the question. And most of the question chances, I am repeating again and again, either you will get question from the formula, there will be a formula, and the option will be the formula given above given formula belongs to the family Fabaceae, Papilionidae, Leguminosae, and all that, right? That kind of, none of above, that kind of, right? So there are only 3 family in your syllabus. So say thanks to God, because there are total 17 family which we study in master classes, and many more subfamily. This is a taxonomy. This is the most toughest part of the biology, taxonomy. So Solanaceae. Solanaceae. Solanaceae is known as the family of vegetables or family of the potato, right? So this is one of the most simplest family, right? Uh, Now let me show you the floral diagram and floral formula. Fabaceae, Liliaceae, Solanaceae. Yes, this is the floral diagram of Solanaceae, right? Now the floral formula of Solanaceae. Let me write it. Now it will be easy for you to understand. This is called family of potato or potato family, or sometime you will come to know that this is also known as the family of vegetables. Family of vegetables. Right. And what is the floral formula? Floral formula is that is actinomorphic. Actinomorphic means this is the what kind of the symmetry? This is the radial symmetry. You can cut down from anyone. The line passing through the radius that will cut down this flower into two equal parts, right? So this is the radial symmetry, right? Actinomorphic. Look, both sex organ are present. These are the androcium, and this is the pistil means gynocium. So that is the actinomorphic bisexual. Actinomorphic bisexual. Calyx 5 gamo. So there are calyx, right? These are the calyx. So sepals are present. All sepals are united. There are 5 sepals. 1, 2, 3, 4, 5 sepals. All are united. So will be calyx 5 gamo. What about the corolla? Can you tell me the formula? 1, 2, 3, 4, 5. Right. There is a 5. So there we can write 5. And all 5 are united means they are gamo. So we'll write it down 5 gamo. Then how many androcium? 1, 2, 3, 4, 5. There are 5 androcium, right? But they are not united. So will not write, will not make bracket, right? They are not united. They are free from. Means they are in polyadelphous condition, right? But you can see that they are united with the petals. Try to understand this one, this line. This line shows that they are attached with the petals. So what we will do, we will make a line. What is line? So we will make a line from androcium to corolla, right? They are attached or they are united with the corolla, right? What we call this line? What is the name of this line? We will call, can you, anybody tell me? When androcium are united with the corolla, we call it epipetalous condition. Epipetalous. This line shows epipetalous means that is, let me write it down, that is androcium or stamen attached to petals. This is called epipetalous secrete, right? Now, what about the gynocium, right? Pistil. So there are two gynocium. There are two half part, distinct part. So there are two half part. So there are two gynocium. So I will write it down. Gynocium 2, gynocium 2, right? And from the floral diagram, you cannot tell it. Epipetal, that is the, these both are united. That is shown over here. Gynocium 2 gamo, and they are ovary superior. From floral diagram, you cannot say that, you cannot tell that it is ovary superior or ovary inferior, right? So this is the floral diagram, right? And what else can be asked to you? The next thing which can be asked, the question can be asked: fruit. What is the fruit? That is also one of the most frequently asked question. The fruit type. So what type of the fruit will be there in this family? The fruit type will be berry or capsule. berry, or capsule. That will be the fruit type. Now, economic importance. Economic importance. I'm just telling you what you can do, right? It's easy, you can do it, right? That's why I'm not telling anything extra for you. But at minimum, what I have written over here, that you have to do at any cost, right? Economic importance. So, tomato, brinjal, and potato, right? So, if we'll talk about the food, it's vegetable, then food, tomato, potato, brinjal, they all belongs to this one. In a spices, chili. Look, potato, tomato, brinjal, chili, they all have same kind of the seed because they belong to the same family, right? So spices, in a spices, chili belongs to this one, right? In medicine, both of the plant have been asked. Medicinal plant come under this family. There is a belladonna. It is applied on the wounds. Belladonna. So belladonna is a medicinal plant. Belladonna tape comes in the market. Nowadays, it is not in that much used, but previously it is used. It was used frequently. You can get it from any particular store, right? So belladonna, and the another one is ashwagandha. Ashwagandha is still used. Ashwagandha. Ashwagandha actually, it's a medicinal plant. It's a root. We extract reserpine from the root, which is used for the blood pressure medicine, right? Reserpine is frequently used nowadays, right? But belladonna is not that much used. It's an older medicine, right? So belladonna and ashwagandha both are the medicine. They belong to the family Solanaceae. You have to remember the name. Then yes, one more plant, tobacco and petunia, right? Tobacco. Remember, tobacco belongs to Solanaceae family. And one more, there is an ornamental plant, petunia. Ornamental plant people uses in garden, that is petunia. You must have seen this plant, petunia. It belongs to this Solanaceae. Tell me if you have any doubt. Go through with it and tell me. Is it clear, Safullah?
Yes, sir.
Uh,
So what's its presentation?
yeah, very good. Presentation. Can you tell me what kind of presentation this is? Let me explain. Placenta is in center. Placenta is at center, but in the same time, there are two margin or septa which divide the family over in two compartments. Tell me. Come on.
Axial.
Very good. Excellent. Placentation is axial. Placentation is also very important. Placentation is axial. Very good. Excellent. Axial placentation. Great. Any more question? So let me ask. Ellen, Eva, Parat, tell me. So are you getting it? Means I'm working hard on it. So is it fruitful or not? Aksa, Hanifa,
Yes, sir.
getting it or just saying yes, sir, in on pressure? No?
No, sir.
Okay.
I get it.
Great. Very good. Now I'm happy. So if you'll do that, if you'll cram, believe on me, if you'll cram whatever I am sharing with you, if you will cram that thing, means suppose that you might be in the regular batch in my medical regular, then also I'll teach you all these things. I have covered all those points which I have covered in the regular batches. I haven't left any single important point, right? So I'm expecting that if you'll go through with it, definitely you can do the same, like you can perform as the same, almost at the same level what the regular batches student can do, right? So don't leave anything or don't take anything for granted. The next family is or the last family is Liliaceae. These all Solanaceae and Fabaceae. These both family belongs to the dicot. Now the family which is the next one Liliaceae. This is the monocot family. Liliaceae. And oh, very close diagram. Look, this is Lily. Every one of you must have seen this one. Very common. You must have given to someone in his or her birthday. So this is the normally people. It's costly. It's a bit costly. But sometimes people give it on birthday and some wedding anniversary and all that and good occasion. So it's a costly, but it's a very beautiful flower. And lily is very look. Here you can see the petals are in two whorl, right? The same floral diagram. You can see the same kind. So the petals are in two whorl, and one whorl is at upside and another whorl is at lower side, right? The same thing have been represented in this floral diagram. You can see, right, in Liliaceae. So this is Liliaceae, right? Look, you can see here Liliaceae. One petal here, this one petal is this one, right? Another petal is this one, another petal, and one petal is this one, right? So this is one word, and another one, this one, and this one. There is another word, right? So you can see that there are no petals. Oh, one more thing, important thing. So one more thing I have to tell you. There are no sepals or petal means there is a no difference between the sepal and petals. So what we will say, right? If the sepals and the petals are not like there is a no difference between the sepals and petals, then what we can see. Uh, so first, this is the actinomorphic. So flowers have radial. This is the radial, right? Means radial symmetry. Radial symmetry. That is actinomorphic. Bisexual, male and female sex organs are both are present. You can see petals and androecium and gynoecium both are present. So bisexual. Then okay. So here petals and sepals are not differentiated. These you cannot say these are petal or sepals. There are only single word, right? Only single word. Either you call it petal or you call it sepal. When we are unable to differentiate between the petal and sepal, right? Let me write it down. What we use the term. When we use the term P. P symbol represent for perianth. Perianth are those when petal or sepals cannot be differentiated. When you cannot differentiate that either they are petal or they are the sepal, then we call it P. We use the term P, and that is called perianth. So what is the perianth? P 3 plus 3, 3 plus 3, and Rusium 3 plus 3, right? So look, there is a no gamo situation. Perianth 3 plus 3, and Rusium 3 plus 3, right? And Gynoecium, again Gynoecium 3. There are 3 chamber. Look, 1, 2, 3 chamber. Gynoecium 3, and that is the ovary superior. This is the actually floral formula, right? And one more important thing, what is the placentation? Can anybody tell me what is the placentation? Tell me, Saifullah, you can tell. What is placentation?
Axial.
Very good, axial placentation, right? So that is the axial placentation. You can tell it from here, axial placentation. And fruit, capsule or berry? Fruit is capsule or berry. This is also called family of lily. Family of lily. And one more important thing which I want to tell you, this is monocot family. Rest two are by dicot family. This is monocot family, right? All the monocot plant come under this family. And what are its economic importance? Let me share with you economic importance of this one. In economic importance, ornamental plant. In ornamental plant, tulip belongs to this family. Tulip. In medicine, aloe belong to this family. Aloe. In vegetable, asparagus belongs to this family. Right. One more important. Colchicine. Colchicine. Colchicine work like gibberellin. It is extracted from this colchicine plant, right? So these are the example. Let me know if you have any doubt. One very important information which I want to share with you. Look, suppose that this is a stem, and flower come out of this system. This is the flower. Flower come out of this one. Right? This is flower is coming out, right? And at the base from where the flower is coming out, the base from where the flower is coming out, there is a small bud-like structure. This is a small bud-like. We call it bract. Bract, maybe it's a leaf-like structure from where the flower originates. Bract may be and may not be over there, right? So bract may be and may not be. If bract is present, we always put br before this one, right? So this flower is called bracteate, right? If bract is present, so br means bracteate. We'll read it. Bracteate. Bracteate means bract is present, right? This is the symbol. This one. This is the symbol of br. In floral diagram, we will represent this symbol. This is br, right? So this is called bracteate. So how we will read this formula? Bracteate, actinomorphic, bisexual, perianth 3 plus 3, and Rusium 3 plus 3, Gynoecium 3 gamo, ovary superior, right? This is the floral formula, and rest of the thing I have explained for you, right? So here is this flower is the bracteate, family of lily. So tell me if you have any doubt.
Sir, can you scroll a little down?
like this way.
Yes. Done, sir.
Right, so this is all about the family. Tell me if you have any doubt. Yes, guys, any doubt? Hanifa, Mamuna, any doubt?
No, sir.
No. So this was all about the that this is the only thing which you have to cram, which you have to remember in this morphology part, right? Now let me show you what is our next topic. Uh, we are already running ahead of our scheduled syllabus. Look, let me show you. This was the schedule. Now today's we are in 26. So 26, there should be chemical control. We have already covered chemical control. We have already covered chemical this plant family part, and now the rest one on 29th we have to do. Sorry, this will take the class. Okay. Oh, Saturday is not my class. So Sunday biology, cell biology and cell cycle. Right. So we'll start with the cell biology part. Fine. Cell biology is also very important. Cell biology and cell cycle. So let me start with the chapter. Go through with it after the class. Go through with it again with the family, and still in case if you are facing any problem, you feel that uh, you have any problem regarding the any question from the family or any concept of the this plant family or morphology part, you are most welcome. Now cell biology is not in this file. Cell structure. So let's discuss one by one. So this is the common diagram of a eukaryotic cell. So here the eukaryotic cell, we have represented a eukaryotic cell with the help of plant cell, right? Here we have discussed the plant cell. Okay. So let me discuss one by one. So cell is the structural and functional unit of the plant. And in 90, this is important. In 1965, there was a scientist, his name was Robert Hooke. Robert Hooke written a book, and the name of his book is Micrographia. The name of the book have been asked multiple times. So you have to remember, the Robert Hooke written a book. The name of that book was Micrographia. In his book Micrographia, he have described the cell. So he is the first person who discovered the cell, right? So what actually the Robert Hooke was? Was he biologist? mathematician, chemistry science, physics scientist, what he was, cell biologist, genetics. Who is the Robert Hooke? Does he belong to biology or not? Don't know. Robert Hooke was the scientist of physics. He did not have to do anything with the biology, right? Robert Hooke was trying to make a microscope, compound microscope, right? Right. As you get the question, who discovered the electron microscope? So who discovered the electron microscope? This question I've been asked. Who discovered electron microscope? Come on, guys, tell me. Safullah, who discovered microscope? Electron microscope. The two scientists named Knoll and Ruska. Knoll and Ruska were the two scientists who discovered the electron microscope. Right. Then the name comes Jacques Janssen. He was also responsible for high resolution microscope, compound microscope discovery of right. So in 1965, Robert Hooke was trying to make a microscope, compound microscope. So while doing the compound microscope, he saw the dead cells in a cork of a bottle, right? And then he further, with the help of that compound microscope, he discovered a number of the dead cells, but he did not see the living cell. After that, a scientist from the microbiology. And the name of that scientist was Antony von Leeuwenhoek, right? Antony von Leeuwenhoek in 1973. Look, after the 8th year, in 1973, Antony von Leeuwenhoek saw the first living cell, and these cells were bacteria, protozoa, and RBC, etc. So you have to remember, if you get the question, who saw the first cell or who saw the first cell, he was the Robert Hooke. But who saw the first living cell? He was the Antony von Leeuwenhoek. So this was a small information which I want to give you. You all know that the outermost structure of the cell is cell wall. It is found in plant only. Then the next membrane, that is the plasma membrane. Inside plasma membrane, there is a gel-like structure that is called the cytoplasm, right? And just cytoplasm. Then there is a large vacuole that is called central vacuole, right? Central vacuole. Then around there are some small structure. These are called cytoskeleton, right? Then there is a structure called chloroplast. Chloroplast. This is chloroplast is one for photosynthesis. We all know. There is a nucleus. One more important thing: vacuole is the largest structure of the plant cell. Nucleus is the second largest structure of the plant cell. But for the animal cell, vacuoles are not found. They are very small or useless and non-functional. So basically, the nucleus is the largest organelle in the terms of in when we talk about the animal cell. Fine. Then around the nucleus, there are is structure called endoplasmic reticulum. Ribosomes are there, which produces protein. Golgi apparatus are there. They also produces enzymes, hormones, and all that, right? Mitochondria, they help in production of energy. Peroxisome, they help in detoxification. They are found in, they oxidize the antioxidant, and they are found in our liver in abundance, right? And they help in detoxification. Plastid, they give the color to the plant, right? So these are the stuff. Okay, this is the important. This is the structure of a bacteria. Now let me discuss with you one by one. Look, the outermost structure, right? The outermost structure of I was making a question paper for a batch. Then I given the question from this one. So the outermost structure of the bacteria. It may or may not be in bacteria, right? So this is a prokaryotic cell. First we have studied the eukaryotic, now this is the prokaryotic. The outermost structure is called capsule or slime layer. Capsule or slime layer, when bacteria lives in water, abundance of the water, or water is available, it will remain a gel-like structure. When there will be scarcity of water or high temperature, this gel will dry and it will become hard. It protects bacteria from the high temperature and adverse situation. This is called capsule or slime layer. That is important. Capsule or slime capsule is made up of a kind of sugar. And what is the name of that sugar? So capsule is made up of a sugar, and the name of that sugar is glycocalyx, right? And glycocalyx is a type of carbohydrate or sugar. Remember, guys, so capsule is made up of glycocalyx, right? It's a sugar, carbohydrate. Then the second layer is cell wall. So bacteria may or may not have capsule. There are fair enough chance that it have, but it may not. If it have the capsule, it will be the tough to kill the bacteria, but if not, then it will be easy. Then there is a second layer that is called cell wall. Cell wall of bacteria is made up of peptidoglycan, means protein plus carbohydrate, right? Peptidoglycan means protein plus carbohydrate. There are two kind of amino acid are found, NAG and NAM, right? So NAG and NAM are two amino acids or protein which are found. Try to understand me. NAG and NAM both have been asked. Peptide cell wall of bacteria is not made up of cellulose like the plant. It is made up of a component called peptidoglycan. Peptido means protein and glycone means carbohydrate. It have a two polypeptide name as NAG and NAM. NAG stand for N-acetyl glucosamine and NAM stand for N-acetylmuramic acid, right? So NAG and NAM are found in this one cell wall. Then there is a plasma membrane. Plasma membrane is made up of bilayer of lipid and protein, right? Then there is a cytosol. Cytosol is made up of water, mineral, and organic substance, right? Then there is a small DNA part that is called plasmid. Plasmid is a small circular DNA, fragment of DNA. And the property of plasmid that is provide the resistance against antibiotic. And we have studied this plasmid in detail in our biotechnology chapter, right? Then bacteria have a chromosome. Chromosome remains attached with a structure. That structure is that outgrowth of the plasma membrane, and this structure is called mesosome. Mesosome work as mitochondria. We have already discussed that thing. That what is the function of mesosomes? Mesosome work as the uh, mitochondria. It help in respiration. And there are some ribosome. We know that ribosome are meant for protein production. The size of ribosome is 70s. Then there are some food granules, that is glycogen. Glycogen is a type of complex carbohydrate. There may be 1 to 4 flagella in a bacteria. Flagella provide it right movement, so it help in the movement. And then DNA, cytosol, we have already discussed. Flagella originate from a root-like structure that is called basal body or blepharum. Right. So you have to remember the NAG, name, glycocalyx, peptidoglycan, right? Stored food and mesosomes. This is the important. Last but not the least, outer surface of bacteria have some small these hair-like structure. They are called membrane. Membrane help bacteria to stuck or get adhere with any of this surface. Tell me if you have any doubt or query in the structure of prokaryotic cell.
So what is NAG and NAM?
Look, this is the full form of NAG and NAM. These are the two. Look, cell wall is made up of peptidoglycan. What is the peptidoglycan? Peptido means protein and glycon means carbohydrate, sugar, right? So basically, there are two compounds. They are basically complex sugar compound, right? They are called NAG and NAM. NAG, the full form of NAG is N-acetyl glucosamine, and NAM N-acetyl muramic acid. Both are part of cell wall of bacteria. Clear?
Okay, sir.
You have to remember the name of both ones. Done? Okay. One more thing. Bacteria, when during the protein synthesis, number of RNA, number of ribosome get attached in a single mRNA, and this structure is called polyribosome. Polyribosomes are found in bacteria, right? They are not found in eukaryotes; they are found in bacteria. So these are called polyribosome. When number of ribosomes get attached with a mRNA during the protein synthesis, and after the protein synthesis, they get detached. So this was the structure of prokaryotic and eukaryotic cell. Now, gram positive and gram negative bacteria. What are gram positive and gram negative bacteria and gram stain? There are two kind of the bacteria, gram positive and gram negative. These bacteria were described by a scientist named as Christian Gram in 1884. Christian Gram stained the bacteria, and with the help of Gram, and that practice is called Gram staining. He taken a bacteria and he colored bacteria with the help of dye crystal violet or gentin violet, that is weakly alkaline dye. So he colored bacteria with the help of crystal or gentin violet weakly alkaline dye, right? So bacteria get blue color. So bacteria get blue color after the coloring with them with the crystal violet, and then bacteria is treated with 0.5% iodine solution, right? Then bacteria washed with the alcohol or acetone. When it is washed with alcohol or acetone, after that the bacteria that retain the blue color, they are called gram positive, and bacteria which become colorless, they are called gram negative, right? But both of the bacteria have to be cleaned with the help of uh, this crystal violet or genetic violet, right? Clear?
Yes, sir.
Now come to the cell wall. Not the bacterial cell wall. Bacterial cell wall we have studied, and I have told you that bacterial cell wall is made up of peptidoglycan. Hai. In fungi, the cell wall is made up of chitin, but in plant, the back cell wall is made up of cellulose. Right? Look, this is the structure of plant cell wall. Now try to understand. This is the nucleus. This is the vacuole, and this is the cytoplasm. Outside of the cytoplasm, there is a layer which is called cell wall, right? And below the cell wall, there is a plasma membrane, right? And between two cell wall, so first there is a primary cell wall, then inner side of the cell wall there is a secondary cell wall. You can get it from here. So try to understand. So first, this part is called primary cell wall. This part, right? This part is called primary cell wall. This part is called primary cell. You can see this is primary cell. This pink color structure, you can call it primary cell wall. So the first cell wall, which is the form primary cell wall, after the when the cell become mature, after the maturity, deposition of substance takes place and formation of secondary cell wall takes place. So this is the secondary cell wall. So inner side of the primary cell wall, at the maturity, deposition of some substance takes place, and when these substance get deposited, formation of secondary cell wall takes place. Right, inner side to the secondary cell wall, there will be plasma membrane. Right, so this was the formation of secondary cell wall. Right, so outside is the primary cell wall, then there is a secondary cell wall. After the secondary cell wall, there is a structure, and that structure is called plasma membrane. You can see this is the structure which is called plasma. This is this structure is called plasma. This third structure, right? This one, right? So this is, and between the uh, two cell wall, there is a substance. Try to understand. Between two cell wall, there is a substance, and that substance is called middle lamella. This is the middle lamella. You can see this structure, this yellow color structure which I have drawn over here. This yellow color structure is called middle lamella. Middle lamella is a cementing substance. This is found between the two cells, and it cement the two cell with the help of uh, cementing material. And the name of that cementing material is calcium pectate. You can see this is middle lamella, and middle lamella is made of all calcium pectate. You have to remember this name because calcium pectate or composition of middle lamella have been asked multiple times, right? So what happened? Uh, uh, Calcium pectate, when I have already told you, when the fruit are unripe, they are very hard because of calcium pectate, this middle lamella. When food get ripened, what happens? Middle lamella get resolved. That's why the ripened fruit becomes soft, right? And the unripe fruit, they are very hard, right? Sometime below the plasma membrane, deposition of some substances takes place at the maturity. This so one more thing, this primary cell wall is made up of very important, very important part. Primary cell wall is made up of cellulose plus hemicellulose plus matrix. Secondary cell wall is made up of cellulose, matrix, and hemicellulose, right? So almost same composition of primary and secondary cell wall. Then sometime due to maturity, when the cell become very older and mature, deposition of some so more substance takes place. They are like tertiary cell wall, right? So sometimes lignin get deposit. Lignin is a permeable substance. Sometimes suberin get deposit. This is fatty substance and it is impermeable to water. And when suberin will deposit, cell will die. Cell will die, right? Cutin is a fatty substance. So sometimes lignin, suberin, or cutin get deposit inner side to the secondary cell wall, which is called tertiary cell wall, right? And suberin and cutin make the cell dead because they are not permeable for the water, right? Due to impermeability of the water, cell die, right? Between two cell, there are some pore. When we talk about the plant cell, between two cell, there are some pore or connections through which exchange of some material takes place. These pores or these channels are called plasmodesmata. They are found in the plant cell. When we talk about the animal cell, between two cells, there are some cytoplasmic interconnections, or they are called cytoplasmic junction, through which exchange of material takes place. Cell wall is semi-transparent, 0.1 micrometer to 10 micrometer thick. They are non-living and metabolically active and can grow, right? This is the property of cell wall. All the point you have to remember. Ask me if you have any doubt. Is that clear? Safula, clear? Ellen, clear? Eba? Tell me, guys.
Yes, sir.
Okay. Now next one, plasma membrane. Now this is easy. Look, plasma membrane is made up of two things. Bi-layer of lipid. Look, there are two layers of lipid. First, I want to tell you that the term plasma membrane, very important. The term plasma membrane was given by two scientists, Nageli and Kramer. They given the term plasma membrane. Nowadays, the cell membrane, plasma membrane, plasmolemma or biomembrane, they all are the same thing. Either you call it cell membrane or called plasma membrane or called plasmolemma or called biomembrane. But nowadays, most frequent term uh, is this one, a biomembrane. Schwann discovered this plasma membrane, but the term plasma membrane was given by Nageli and Kramer. Right. What is the composition of plasma membrane? Plasma membrane is made up of two things. Bi-layer of lipid means two layer of lipid and a protein, right? Protein. So look, there are this one, and this is second. There are two layer of lipid, and between the lipid, there are some protein, blue colored structure. They are called protein. This structure of the plasma membrane was given by two scientists, Singer and Nicholson. They said that what is the state of plasma membrane? Plasma membrane is fluid, it's a fluid mosaic model. It is arranged like a fluid and mosaic. They said that this lipid work like a fluid, and this one lipid work like a fluid, and this protein they work like integral protein or some mosaic like structure, right? So Singer and Nicholson in 1972 given concept that this is a fluid mosaic model. It is a quasi fluid, means not completely fluid or semi fluid, quasi fluid, right? It is there are some enzymes which are called permeases. We'll discuss in later. Try to understand this. Bi-layer of lipid, there are two layer of lipid. Now you can see there are lipid, there are yellow color head and there is a black color tail, right? This yellow color head part, it always remains at the outside. This head part is hydrophilic part, right? And the tail part is hydrophobic part. Hydrophilic means water loving part. Hydrophobic means water repellent part. So this central part is hydrophobic. It is called hydrophobic tail. It is written over here. It is called hydrophobic tail, and this upper part, yellow part, is hydrophilic head, right? And in between there are some blue color protein. These protein which traverse across the membrane, they are called integral protein. Those protein which are found at the surface, they do not traverse across the membrane, they are called peripheral protein, right? Between in this protein layer, there are some channel protein, they are called channel protein. They are channel protein, right? These channel protein are gateway through which ions and iron and water or some substances go through across this one, right? We have already discussed this channel, uh, uniport, symport, antiport, and aquaporins. These are those channels which we have discussed over there. They are ion channels or aquaporin, right? And we call them channel protein, right? So this is the structure of plasma membrane. Tell me, is it clear or not?
No doubt, sir.
No. Very good. So remember who given the term? Kramer and Nageli. That question I've been asked. Fluid mosaic model was given by Singer and Nicholson in 1972. They got the Nobel Prize for the same. That also you have to remember. One more thing which I have written over here. The percentage of lipid may be 20 to 79. Percentage of protein may be 20 to 79. Carbohydrate 1 to 5% and water 20%. Right. The percentage of lipid and protein that vary from cell to cell. Otherwise, the structure of plasma membrane or any kind of the membrane will be same across the earth. Across the earth. Either it's protozoa or it's bacteria or it's anywhere else. Right? So plasma membrane composition will never change. Only the ratio of protein and the lipid will change. Now the protoplasm. You have not to remember everything. You have to remember only those things which I have underlined with the red. Cell have two components, cell coat and protoplasm. Cell coat may be cell wall or capsule. Capsule is made up of glycocalyx, bacteria and animal cell. And cell coat is made up of cell wall. Again, protoplasm divided into three parts. Cytoplasm, cell membrane, means plasma membrane, and nucleus. Right? Further, cytoplasm is divided into two parts. Hyaloplasm and cytoplasmic structure. Hyaloplasm is also known as the cytosol, right? And cytoplasmic structure or mitochondria, Golgi complex, and all that, right? So that is the cytoplasm. So all the gel-like substance which is found inside the plant, this is called cytoplasm. So let's discuss organelle one by one. This is the structure of endoplasmic reticulum. ER, endoplasmic reticulum. Most of the scientists says that endoplasmic reticulum are the extension of the envelope of nucleus membrane, right? So they are the envelope of nuclear membrane, right? Folding of nuclear membrane. So because they remains interconnected with the nuclear membrane. This is the red color nucleus. Then this blue color structure is nucleus and nuclear envelope. Then there are outer side there are four nuclear core, and this folding is interconnected with it. These are rough endoplasmic reticulum because the ribosomes are attached outer its outside surface, right? They are hollow pipe-like structure. So endoplasmic reticulum are of two types: this SER and RER. SER are smooth endoplasmic reticulum. RER a rough endoplasmic reticulum. A smooth endoplasmic reticulum, their function is they produce. I have written directly over here. They produces lipid and steroid. This question will be asked. Right. I have seen this question. What is the function of SER? What is the function of RER? That kind of the question you will get. In fact, they have already been asked. Right. So, look, the smooth endoplasmic reticulum, their function is to produce lipid and steroid. Steroids are a type of hormone. Rough endoplasmic reticulum, they have ribosome on their surface. Right. They produces enzyme and protein. Right. They were discovered by Porter and Thomson in 1945, right? The space inside the these structure are called lumen of endoplasmic reticulum. Very important fact: they are not found in egg and mature RBC. They are not found in egg, mature RBC, embryonic cells, prokaryotic cells, etc. Right? Ribosomes remains attached. This is also very important fact. Ribosome remains attached with the surface of endoplasmic reticulum with the help of protein called ribofourin 1 and ribofourin 2. Rough endoplasmic reticulum (RER), they are uh, they uh, uh, they are they produces the precursor of enzyme or precursor of lysosome and Golgi complex. Means the Golgi complex and the lysosome, they originate from the rough endoplasmic reticulum. SER, they senses the fat found in adipose tissues, They produces glycogen in the liver tissues. They produces fat in adipose tissue. They produces glycogen in liver tissues. They synthesize steroid hormone. That is the function, right? Sarcoplasmic reticulum are a type of endoplasmic reticulum. They store the calcium, and we have studied this sarcoplasmic reticulum in sliding filament theory, right? Tell me, go through with it and tell me, clear or not? Done. Parat, is it okay? Alan, Aksa,
Yes, sir.
Hiba.
Yes, sir.
Okay. Then come to Golgi complex. Again, Golgi complex. Look, Golgi complex are this kind of the structure. They are also known as the Dalton complex, right? And look, there is a phase, cis phase. Cis phase is found toward the nucleus, and this phase is called trans phase, right? Trans phase remains toward the cell membrane, right? So this is the polar organelle. First, very important. Uh, there are these kind of the structure. These are called cisternae. These are called cisternae. So what happened? The protein come from there, from where? Rough endoplasmic reticulum. So protein come from a rough endoplasmic reticulum, and they modify the protein. They pack a protein in a vesicle and they secrete outside. So secretion takes place toward the transphase. This one, transphase. Secretion always takes place toward the transphase. These secretory vesicle come out of. So what is the function of Golgi complex? Packaging of protein. From where the protein is coming? From rough endoplasmic reticulum. Right. This Golgi complex make three kind of the packet of protein. Three type of the packet of protein. First, A, B, and C. Three type of the protein packet. First kind of the packet, they go and fuse with the plasma membrane, right? And they help in the formation of plasma membrane. And this process is called biosynthesis. Formation of new plasma membrane is called biosynthesis. So when the new plasma membrane is formed or the old plasma membrane get damaged, then the protein is supplied by Golgi complex. So they send a vesicle filled with the protein and help in the repair or formation of new plasma. Second, B kind of the vesicle, they come out of the cell and they work as an enzyme. C kind of vesicle, they come out of the they remain inside the protoplasm and they form the lysosome. So formation of lysosome takes place from the Golgi complex. Golgi complex were discovered by Camillo Golgi and it named after his name. It was discovered. This is very important question. This question I've been asked. The Golgi complex have been discovered in the nerve cell of brine owl or cat, right? Except sheep tube of plant, sperm of bryophyte and pteridophyte, and RBC of human. They are found everywhere. Unconnected. So one more thing. I have a question. Golgi complex are found in plant or not? Tell me. Golgi complex are found in plant or not?
Yes.
They are found in plant for sure, or something else is found. Guys, Golgi complex as such, they are not found in the plant. In the plant, we find the same Golgi complex. Yes. If someone says that Golgi complex are not found in the plant, then that is a wrong notion. But if someone says Golgi complex are found in the plant, that is not exact answer. Look, these kind of unconnected cisternae are found. This kind of a structure, unconnected cisternae are found. They are known as the dictyosome, right? So in place of Golgi complex in plant, dictyosomes are found. They are the collection of 3 to 20 unconnected cisternae, right? So dictyosomes are found in place of, while they have the same function, in place of Golgi. In place of Golgi in plant. So plant have dictyosome in place of the Golgi complex. Clear? And what are they? They are the unconnected cisternae. Very important. Lysosome. Questions are being asked from lysosome as well. Lysosome, we have already discussed that the formation of lysosome takes place from the Golgi complex. Their outer structure, outer membrane is, they are made up of single membrane, right? Golgi complex are made up of 50 different type of the enzyme. They are known as the suicide bag. Hydrogen ions, they pump in hydrogen ion, and these hydrogen ion increases the acidity, right? Right? These are the channel protein, right? Or pump of hydrogen ion, right? Channel protein or pump of the hydrogen ion, and they are known as the scavenger of the cell. Discovered by Christian Dudue. pH value is 4 to 5. They are found in all animal cell except RBC, right? Red blood corpuscles. Plant and fungi, their function is taken away by the vacuole. Means in the plant and fungi, they are not found. Their function is taken out by the vacuoles. And Golgi complex have, this lysosome have very strong hydrolytic enzyme, right? Very strong, that you have to remember. Very hydrolytic enzyme, strong hydrolytic enzyme in their lumen, their image film. Their function is digestion of food, nothing else, right? And their diameter is 0.2 to 0.8 millimeter. Look, what is their function in prokaryotic cell? Try to understand. Cell eats something that is called phagocytosis. When phagocytosis, through phagocytosis, it form a food vacuole. This is an amoeba, right? It have engulfed the food vacuole. When food vacuole will go, it will, this Golgi complex will, this is lysosome. So I am sorry, lysosome. This lysosome will fuse with this food vacuole. They will digest the food. The digested food will be excreted out, right? And useful substance are um, utilized, right? So that is the function. Tell me, is it clear? Lysosome. They are called suicide bag. Why? If they rupture, the complete cell will die. They are called scavenger of cell. Why? Because they eat and digest old or dead cell organisms. Who discovered it? You have to remember. What type of enzyme are found inside it? You have to remember, right? Where they are found or where they are not found, that also you have to remember. Tell me, guys, is it clear or not?
Yes, sir.
Fine. Vacuoles. Vacuoles are basically well developed in the plant. An animal where they are very small and there is a very minimal function. And in mature plant cell, they are found in mature plant cell. They are the largest organelle I have told you in plant cell. Protoplasm, nucleus becomes peripheral because vacuole become large and it push the nucleus and protoplasm toward the periphery. The membrane of that is important thing. Membrane of vacuole is known as tonoplast. It is selectively permeable membrane, right? So membrane of vacuole is known as tonoplast. Vacuole remain filled with the water, pigment, minerals. They maintain the shape of the cell, right? The pigment which is also stored inside the vacuole is called anthocyanin, right? Some protozoa have contractile vacuole, right? This is the plant vacuole. This is the central vacuole, right? But some animal cell have contractile vacuole, like amoeba and paramecium. The function of this contractile vacuole is excretion. Tell me. Is that clear? Vacuole, very simple. Remember the function of contractile vacuole. Contractile vacuole are found only in animal cell. Mitochondria. The structure of mitochondria we have discussed in detail in that part, that respiration part, but still I will remind you. There is a outer membrane, inner membrane. Inner membrane is folded. This folded structure are known as the cristae. We know that mitochondria have its own DNA called mtDNA. This is semi-autonomous. It is circular DNA like bacteria, and this resemble with the bacteria, right? The inner folding of mitochondria, inside the mitochondria there is a matrix, the air, and it have ribosome, 70s type ribosome. Inner folding of mitochondria, and in this matrix Krebs cycle or tricarboxylic acid cycle goes on, right? These folding are known as cristae. Inside the cristae there are some these lollipop like structure. This is called oxysomes. The oxysome, this is again the structure of oxysome. The base is called F0 particle, the upper part is called F1 particle. The upper part, in the upper part is made up of ATP synthase enzyme. Formation of ATP takes place in oxysome head, right? And this reaction is called oxidative phosphorylation. Oxidative phosphorylation is formation of ATP from ATP. Right? Krebs cycle, we know burning of the food that take place in the matrix part. Remember or not? Come on, guys.
Yes, sir.
Okay. So mitochondrial structure we have discussed in detail. We need not to discuss. Even chloroplast we have discussed in detail. Plastids, that is important. That you need to remember. You can get question from. Look, plastids are found only in plant. They are made up of proplastids. That is the immature plastids. Plastids can be of three types. Chromoplast, they have pigment and they give color to the flower and fruit. Chloroplast, that is meant for photosynthesis and lipid synthesis. Leucoplast, synthesis of fatty acids and amino acid takes place, and its function is the storage of food. Further, the leucoplast can be of three types: amyloplast, elioplast, and proteinoplast. If leucoplast is storing the sugar or starch, that is called amyloplast. If it is store oil or lipid, or elioplast. If it store protein, called proteinoplast or proteinoplast, right? So that's it for the day. In next class, we'll start from the chloroplast onward. Tell me if you have any doubt. I think this chapter might be easier for you because we have covered most of the part in different chapters. Tell me, students.
Yes, sir.
Okay. So see you next class. Bye bye. Take care.